So welcome everyone. Good morning. Thank you for being here today. Welcome to one of our webinar series for 2024. This is Dr. Kath Skopelitou and I will be your host for today's webinar. So before I will introduce you to our speaker, let me tell you some more things about EU-OPENSCREEN. EU-OPENSCREEN is a European research infrastructure consortium for chemical biology and early drug discovery. We support all stages of a chemical to development project, including assay adaptation, high throughput screening, and chemical optimization of the hit compounds. Furthermore, EU-OPENSCREEN operates an open access database and a unique common compound collection. Our main library, the European Chemical Biology Library, consists of over 100,000 compounds. EU-OPENSCREEN has over 30 affiliated high-throughput screening and chemistry facilities at partner sites in 9 European countries. These partner facilities provide researchers with access to cutting-edge technologies to develop their own tool compounds. If you would like to know more about our training activities, you can always navigate in our website. You can go to Participate tab and Access to Training, and there you can see all of our training activities. And if you would like to receive all training activities directly in your email box, you can contact us at training@euopenscreen.eu. A few more things about our webinar today. At the end of the webinar, you can ask your questions directly to our speaker by raising your hand, or you can just type your question in the Q&A box at the bottom of your screen. So now it's time to introduce you to our speaker for today, Dr. Snežana Đorđević. With a background in pharmacy and analytical chemistry, Snežana dedicated her research career to the multidisciplinary investigation in the nanomedicine field. She's currently a postdoctoral researcher at Research Center Principe Felipe, and her contribution to the nanomedicine field is focused on increasing awareness of precise and exhaustive physicochemical characterization of new drug delivery materials. As mass spectrometry enthusiast, Snežana will give a talk on mass spectrometry imaging and its implementation in biodistribution studies of polymer therapeutics. Snežana, if you are ready, we are very eager to hear to your talk. I will now stop sharing my screen.
Thank you very much. Let me... Yeah, can you see my screen?
Yes, we can see your screen now.
Let me just put it in the... Okay.
Great. That's great.
Great.
Thanks.
Okay, good morning, everyone, and thank you very much for being here today. And I have to give a great thanks to EU-OPENSCREEN for organizing this webinar, in which I will try to transmit my enthusiasm for mass spectrometry imaging and its implementation in polymer therapeutics field. So before starting introducing you to nanomedicine and mass spectrometry imaging, I would like to give you a challenge. Try to think of a significant scientific advancement that was not made possible by the development of a tool that enabled us either to see or measure something. It's struggling, right? Well, the invention of mass spectrometry had a great and a significant impact in those advancements. At first, MS were large, clunky instruments that were not computerized at all. But now, as a result of significant technological advances, MS has become a golden standard for solving problems in a wide range of fields, and including the nanomedicine where I work in. So, first of all, I would like to introduce you to nanomedicine, a field where the nanotechnology-based strategies aim to improve the water solubility of small drugs in the bloodstream and then promote their transport across biological barriers, but also to prolong circulation times, meaning that we have the longer time between the doses, and we can control the timing and the location of therapeutic release. All of this can lead to increased safety and efficacy of active moieties. So how is this possible, you may ask? Well, by the use of implementation of the enhanced permeability and retention, a phenomenon that relates to the anatomical architecture and pathophysiological processes in the disease tissue, applicable to either cancer or inflammation or infectious diseases. So compared to the healthy tissue, the tumor or inflammatory tissue is characterized by the overexpression, for example, of inflammatory factors, then irregular vascularization or disruptive walls of blood vessels. But apart from that, we can have also the lack of lymphatic drainage that leads to passive accumulation of nanomedicine. You probably heard about nanomedicine and liposomes a lot lately due to the COVID-19 vaccine. But apart from these liposomes that are used to deliver mRNA vaccine, the nanomedicine includes many other groups, such as lipid nanoparticles, polymeric particles, polymeric micelles, polygroup polymer therapeutics, nanogels, hydrogels, and etc. And the application of these groups may differ on a case-by-case basis. And with every group can offer specific advantages, but also suffer from various disadvantages. And as you can see here on this timeline, since their first appearance and approval in the 1980s, nanomedicine have fundamentally changed how we address unmet clinical needs, including, but not limited to, the treatment of cardiovascular, lung, kidney disease, but also neurodegenerative disorders, and mostly focusing on the primary and metastatic tumors.
Snežana, sorry for the interruption. We have a small problem with the sound. Maybe you can use your headphones or... Yes, we have a bit of a problem.
You cannot hear anything, or?
No, it's just a small problem. We can hear you, but not clearly enough.
Vale, let me try to get my...
Thank you so much.
Yes. Technology, right?
Yes.
Let me just...
Maybe the sound will be fixed now.
Vale, what about now?
I think it's much better now. I think it's much better. Yes.
What about the others?
I think we have a chat, so if there is a problem, I will interrupt you again.
Vale, okay, good. Thank you very much.
Great, thanks.
So, where I was. Okay, so in our lab, we focus mostly on the use of bioactive polypeptides and polypeptide drug conjugates from the Polymer Therapeutics panel, which means that in this case of the polypeptide drug conjugates, the drug is conjugated to a biodegradable polyglutamic acid through a bioresponsive linker. These conjugates are supposed to be stable in the bloodstream since they are connected through a bioresponsive linker. And once they reach in the tumor, the drug is released either by the influence of the low pH in the tumor, then the high glutathione concentration that breaks the redox sensible reductive sensible linkers, or the presence of the specific enzymes, or even the presence of cathepsin B. A great advantage that we have in our Polymer Therapeutics lab is the possibility to perform both the synthesis and the biological evaluation in-house in our lab. But in the last, let's say, 5 or 6 years, we added a third, but also essential part of every lab, which is analytical chemistry. Meaning that we are integrating analytical chemistry concept by developing the fit-for-purpose analytical procedures for the characterization of our systems. And during my PhD and now as a postdoc, I have been leading the analytical chemistry part of Polymer Therapeutics lab, meaning that I have been involved in physicochemical characterization of almost every system design in our lab, but also some of the EU-OPENSCREEN projects. But the main focus, let's say, of my role is to adapt already existing and develop new protocols that will help us in understanding better the in vivo behavior of our PGA drug conjugates. And that someone, as Katie said, who is MS fanatic, the mass spectrometry imaging represents one of my favorite strategies so far. So let me tell you why. As you could conclude from, let's say, from the introduction parts, in the introduction slide of my of the nanomedicine field, we can conclude that the biodistribution and the delivery of active moiety or the drug to the target sites represents a crucial and critical issue for nanomedicine, especially since this property directly relates to their efficacy. Meaning that we must undertake a proof of principle studies early in the development stages, and all of that in order to demonstrate the delivery of the active drug to the site of action, but also at the necessary concentration. The LCMS, as an extremely sensitive and selective method, is definitely considered as a standard quantitative and qualitative analytical tool for this purpose. However, the loss of, during the implementation of LCMS, we can lose the information regarding the spatial distribution because we are actually extracting the drug from the tissue and analyzing it qualitatively and quantitatively. But apart from that, we can also lose the spatial distribution of the metabolites in the tissue, which represents one of the really significant disadvantages of LCMS. So apart from LCMS, alternatively, we can use some imaging technologies that provide methods which compensate for this disadvantage of LCMS. However, these methods, again, now have some disadvantages, and they're already established. Even though that they are already established protocols, such as, for example, for autoradiography or fluorescence imaging, they suffer from other problems, including, for example, the need for radioisotope labeled compounds or fluorescent labeled compounds, but as well to difficulties to distinguish the administered compound from the metabolites that are internally available in the tissue. Additionally, apart from this, the fluorescence studies that we are now performing for the biodistribution are mostly performed on just the carrier and not following the drug itself. So I cannot help but ask, what if the dye changes the biodistribution of the whole entity, or it affects the interaction of the nanomedicine with the biological environment? Then what if the fluorescence studies gives the false positive results? Meaning that what if they show that the carrier arrived to the tumor, but in the reality it didn't? Or what if the drug actually arrived to the tumor, but not with the carrier? But let's say, for example, now let's forget all of those questions in the beginning and say that it arrived together with the carrier to the site of action, but it wasn't released. And what if it was released, but the tumor distribution is not homogeneous? What, right? Then how does all of that amount of the carrier affect the metabolism that is present in either the tumor tissue or the healthy tissue? And last but not least, is the metabolism of the drug changed? So to all of these questions and many more, we hope to be able to answer with mass spectrometry imaging, more specifically multi-mass spectrometry imaging. I'm sure that you have already heard about multi-mass spectrometry, but mostly, I'm sure, in the context of metabolomics, proteomics, and all of the other omics studies in science. But omics is aside, MALDI is also used as an imaging technique and even as a called as a label-free imaging technique. How can we perform it? Well, I'm sure that you know that each molecule that we have, either a metabolite or a lipid or even a drug, possesses a specific molecular weight, right? So if we can determine this weight, we can identify that molecule. So how can we determine this molecular weight? By using mass spectrometry and by sending the molecule, let's say, out on a race and measuring their finishing time at the detector. But first, how are we going to generate now the molecules from these molecules from our samples? Let me illustrate that in here. So we can use in the MALDI, it's used that we use a laser. And in the MALDI-TOF, we can, for example, use the multi-laser photons and accelerate them towards the surface of the sample to generate so-called the molecular splash that will carry all of the analytes and the tissue fingerprint that now afterwards enter the MS analyzer, where we can determine exactly the molecular weight of these compounds. So we can do this point by point on a given sample and create a molecular images that looks something like this. So in this image, you can see that each image separately represents a local distribution of one specific molecule on the same sample and analyzed just once. So we can say that from all of this, that a well-designed MALDI MS imaging experiment usually contains 3 steps. The first one is the sample preparation, where the sample is collected, sectioned, and coated with the matrix that can absorb the energy from the MALDI laser, so-called MALDI matrix. Then the mass spectra acquisition, where the MALDI matrix coated section is irradiated by the MALDI laser beam to ionize the analytes that are present in the sample, and then that they are analyzed in the mass analyzer. And in our case, as I explained, is the time of flight of TOF analyzer. The last part is maybe the longest one, is the data processing and the data analysis, where the intensities of the specific mass to charge ratio values are processed and plotted with the specific spatial information, and where we can use a color gradient to create a MALDI MS image. So now that we all know how to perform a MALDI MS imaging experiment, that leads me to our first project and my first example of the use of MALDI imaging, where we aim to investigate the potential of this technique to evaluate the drug release and the spatial distribution in 3D triple negative breast cancer cell lines after the treatment with combination PGA drug conjugates. What does this mean? It means that we synthesized and designed first the combination PGA drug conjugate, where we connected and conjugated basically 2 drugs to the same polyglutamic acid chain through a disulfide redox responsive linker. Then we used this PGA drug conjugate to treat the 3D triple negative breast cancer cell lines, where we wanted to actually see where the drug was released in those in vitro models. I have to mention that we performed this project in a collaboration with Sheffield Hallam University, where we performed all of the MALDI imaging experiments, but everything else we synthesized in our lab and as well as in vitro studies. So the samples that we used were 2 different in vitro models. 3D, both of them were the 3D models. A 231 triple negative breast cancer spheroids and mammospheres, where each are represented here, as you can see on this scheme, by yellow circles. So the first step, as I mentioned before, is the sample preparation process. And this sample preparation process can differentiate between the sample types. However, there are, of course, some basic principles that need to be considered in each case, such as the sample collection, the sample sectioning, and ionization aiding treatments. Usually, when we perform the multi-MS imaging, the best results we get from a fresh snap frozen tissue that can be either achieved by different solvents, depending on the sample type. For example, depending on the size, depending on the fragility of the sample. So as you can see here, in our case, we use the 3D cell models, a very fragile samples. So in order to conserve the sample integrity, we froze them in the isopropyl alcohol that was previously chilled in the liquid nitrogen. So not indirectly in the liquid nitrogen, but actually using the isopropyl alcohol that was chilled in liquid nitrogen. In that case, in this sense, we can actually perform freezing of our tissue, of our samples without damaging it, because of the freezing is not as fast as if it would be in the liquid, directly in the liquid nitrogen. This protocol, by the way, was also performed and developed at the Sheffield Hallam University. And after the freezing of our samples, they section them at 10 or 20 micrometers, a thickness that is comparable to a mammalian cell. So in this way, the majority of cells are cut open and the multi-matrix is allowed to co-crystallize with the content of the cells. So next step in the sample preparation process is the application of multi-matrix solution. This multi-matrix solution can consist of multi-matrix, of course, where and some organic acid can be used to absorb the laser energy in the by the multi-laser, leading to the explosive desorption of analytes that are held within the matrix signals, matrix crystals. Sorry. The second part is the organic solvent, in which we can use either the methanol or acetonitrile, and is used for dissolving, first for dissolving of the multi-matrix, but on the second part, it's also its role is to rapidly extract the analytes of interest that we have in our sample, because it's evaporating, and allowing their co-crystallization with the growing multi-matrix crystals on the sample surface. Which means that afterwards, when we have the multi-laser and ionize the analytes, they are ionized by the help of multi-laser, multi-matrix. The third component is the strong organic acid. So, for example, if we are performing the positive mode ionization, we can use a strong acid such as trifluoroacetic acid that will increase the analyte ionization by enriching the surrounding media with protons. So, this matrix choice and the matrix solvent choice is mainly driven by the type of analyte, as you would imagine. So that we need the analyte that we want to image. So it can be changed depending if it's a drug, if it's a lipid, if it's a protein or peptide. So all of that can be optimized for your specific case. In our case, we found that CHCA is an optimal matrix that we dissolved in methanol and with the addition of trifluoroacetic acid because the drugs were basic and we needed them to be ionized. So this matrix solution enabled us to obtain the highest relative intensity of both drugs and to image their distribution on the spike samples. So in here, in these figures, in the first ones, you can see that we obtained the signal of both our drugs that we spiked our samples, our 3D in vitro models with. However, one of the things that we noticed is a slight interference for one of the drugs in the tissue. Because as you can see in the control samples of the first drug, drug 1, the surface is everything is blue, meaning that there is no signal for that molecular weight. However, in the case of the drug 2, we can see that there is a slight interference of the matrix, in this case the tissue, that will interfere our results afterwards for the drug. Apart from that, we also were able to observe the ion clusters. Something that is happening when a drug molecule is connected to a multi-matrix, so those clusters, we can actually see them in the MS analyzer. But in this case, we suggested that this was happening because we dissolved the drug in the matrix solution, which means that those clusters happen, let's say, because in the solution, and then afterwards, when they were ionized, we could detect them. But this doesn't mean that we will see these clusters in the real samples, because the drug should be already present in the tissue. So after all of the preparation and using the matrix and the matrix solvent, we initiated our multi-top imaging. And once you start the multi-top imaging, you have to first perform the profiling of the control sections, meaning that we performed, for example, the imaging in the positive ion mode in order to demonstrate the compatibility of our in vitro models with mass spectrometry imaging. In other words, we just wanted to make sure that we can see the spheroids and the mammospheres by MALDI-MS imaging. And indeed, as you can see here on these images, we were able to obtain the multi-molecular images of our in vitro models even at 2 resolutions, so at the 10 and the 30 micrometer resolution, and with lipids such as on this big image, you can see it's phosphorylcholine, and the choline head as primary metabolite, meaning that they were the ones that were the most abundant in the MS spectrum. In this image, each spot represents a specific and individual spheroid or mammosphere, depending on the model, whose localization then we confirmed by H&D staining. However, the conclusive identification and the detection of the drugs in our in vitro models was proven to be a little bit problematic, with one of the reasons being actually the high limit of detection of multi-top instrument at Hallam University. So what we did, we increased the dose of the treatment of our PGA drug conjugate in order to see if we can improve the detection of our drugs. However, that didn't prove to be helpful. But as it usually happens in science, the ideas and the solution to our problems happen when you least expect it. And for us, the light bulb started glowing once we finished another project, a parallel project that we recently published, the linker study, so that we call it. And then we actually performed it at the same time and mostly after the MS imaging experiments. And in this study, what we found is actually that we observed around 40% internalization of our conjugate, and only 0.32% of those 40% released the drug in the 3D mammospheres. So we can conclude that there was actually insufficient drug release in the 3D in vitro models in the first place. So it was obvious that we couldn't detect it with MALDI-MS imaging. So all of these results just reinforce our suggestion that we need appropriate techniques and we need the adequate protocols for the characterization of our conjugate, for the drug release, for the mimicking the drug release, and to perform them in the cells and not just mimicking it in the solution, and not to be afraid to keep the old protocols just because we didn't want to question the already established, actually to improve each year. But as even though that we didn't detect the drug that was released our conjugate. As mentioned before, at the same time, MALDI is collecting all the MS spectrum ions during the same experiment. So we could do some other studies, which is the metabolomics and lipidomics as well, that we analyzed, and we analyzed the metabolomic fingerprint of our triple negative breast cancer in vitro models. As you can see on these PCA plots, we observed a clear metabolomic difference between untreated spheroids, which are the green circles, and those ones that were treated with the PGA, one of the PGA drug conjugate, and the free drugs. We obtained the same results in the mammosphere model as well. However, one of the conjugates, it's actually this one from Fasudil from our linker study paper, and the combination conjugate demonstrated similar metabolomic signatures as the control sample, confirming that we have confirming the low toxicity that we actually observed in the spheroid model. But as opposed to spheroids, the PCA plots of mammospheres clearly differentiate the metabolomic signatures between the Fasudil conjugate and the combination conjugate compared to the control samples, which all of that relates to the higher toxicity in the mammospheres compared to the spheroids. And one of the things that we suggested that the reason behind these results are mostly because of the higher GSH glutathione content that was present in the 3D mammosphere compared to the spheroids. Because if you remember at the beginning, the introduction slide, I mentioned that we designed these conjugates by conjugating the PGA through a redox responsive linker, disulfide, that is cleaved by the GSH. So the 3D mammospheres that have the higher content of the GSH will lead to the more toxicity and more release of the drug and more toxicity. A second example that I will talk today about you today is our implementation of MALDI MS imaging to evaluate the drug release again and the spatial distribution of another drug, paclitaxel, that is conjugated to a star-shaped polyglutamic acid after the intranasal administration in an orthotopic in vivo mouse model of glioblastoma. Such a long sentence, right? But it makes sense. So this time, as you can see, we wanted to check first another drug. We wanted to check the intranasal administration, and we wanted to check if we can use MALDI-TOF for imaging tumor tissues, tumor and the healthy tissues. So instead of in vitro models, in this case, in this project, we had brain tissue as the sample. But as from all of the results, the previous results that we obtained in the previous study, that as well, since we knew that the goal of our project was actually to see the tissue distribution of paclitaxel that was released, only the one that is released from its conjugate inside the brain, one brain tissue section at a certain time point, 1.5 hours, in this case, at 24 hours, we knew that we are dealing with extremely low drug concentration, which is why we needed to take out basically our big guns. And big guns in this case, in the case of MALDI, is the MALDI-2 technology that where apart from having a basic MALDI laser, there is another MALDI-2 ionization source used as a second laser that is parallel to the sample surface and that fires into the already evolved gas of ions, meaning that the molecules that are already in the gas that were not ionized, the MALDI-2 will do the transforming. It will transfer the charge to those molecules, and we can increase the sensitivity of our method, and we can reduce the ion suppression effects that we can have from our matrix. And indeed, as you can see here, we first wanted to compare the MALDI and the MALDI-2 laser, if it's useful for our purpose, or if it makes sense to use the MALDI-2 and not just MALDI. And as you can see here on this graph, we obtained significantly higher paclitaxel signal with MALDI-2 laser, orange bars, and compared to the single MALDI laser, that is the blue bars. And we obtained these results, the higher intensities, in both the standard solution and in tissues that we spiked with the standard solution of our drugs, both the healthy and the tumor one. But the only difference that we saw is the difference in the adduct ions of paclitaxel that we could detect. In the case of the standard solution, we could detect more the sodium ion of paclitaxel. Why? Well, because if you know that the glass slides of the MALDI that we have and that we use for the MALDI imaging, the glass is filled with sodium ions. So there is a high abundance of sodium ions of those glass. So it's normal and logical that we will see more sodium adduct ions in the standard samples, because we just used and spotted the standard solution directly on the ITO slides. But in the case of the tissue spiking, we spotted the standard solution on the healthy tissue and on the tumor tissue. And in the tissues, we have apart from sodium, we also have potassium ions. So it was normal that we could see as well the potassium ions. However, all of this, as previously in the previous project, as I mentioned, these are not the real conditions. And this doesn't mean that we will detect only potassium adducts in the real samples, since we cannot mimic the real conditions. We cannot mimic completely the release of the drug directly in the tissue unless we have the real samples, which is why in the next step, we needed to follow all of these 3 adduct ions in order not to miss anything. So when it comes to the real samples, here, as you can see, we could detect the sodium adduct ions of paclitaxel and not the potassium that was released from its conjugate, meaning that this is the first example where the MALDI-2 technology supported us with the detection and the localization of paclitaxel that was released only in the tumor region. And on the left side, you can see that we detected a higher signal 24 hours after the intranasal administration. And these results confirms that we were able to deliver paclitaxel to the tumor, to the site of action, with our STAR-PGA conjugate. But now the ones who have experience with the MALDI or with the mass spectrometry would ask, how are we sure that this paclitaxel was released in the tumor, in the tissue, and not fragmented in the MALDI source? Well, we analyzed also the standard solution of both the paclitaxel, STAR-PGA, and of our conjugate on the same slide where we have this sample. So we spotted the sample solutions, and as you can see, we couldn't detect the paclitaxel, neither the sodium ion, neither the potassium, or just hydrogen ion in the standard solution of STAR-PGA conjugate, meaning that this paclitaxel that we detected in the real sample is actually the one who was released. So now that we had paclitaxel covered, we wanted to check if we can localize the STAR-PGA as well. However, that part was proven to be a little bit problematic, as expected. But even though that we are still in the process of developing of the protocol, I can show you some preliminary results where we found, what we found really interesting is that we were able to detect not the STAR-PGA as a whole, because it has a really high molecular weight, but actually the STAR-PGA fragments. And that we could detect by using the fractional KMD plots. And these results just only suggest that we can sometimes the unnecessary analyte fragmentation in the multi-source can actually be useful for certain studies. What made our study even more interesting is that we found a different spatial distribution of the detected PGA fragments, these fragments of the star PGA, with small and the bigger molecular weight. As you can see, they had a different spatial distribution in the different parts of the brain tissue, depending if it's a high molecular weight fragment or. Furthermore, as you can see here on this image, on the right one is the MS imaging image, and on the left is the MRI image of the glioblastoma in one of the mouse. We could perform this image of the multi of the right is the segmentation analysis of our brain cells, where each color represents a specific metabolomic signature that in this case clearly differentiate the tumor from the healthy tissue, then the metabolomic heterogeneity inside the tumor, but as well even the migration of some cancer cells to other parts of the brain. But this we cannot be sure that it's that this is the case of the migration. It can also be just the changes, the slight changes, metabolomic changes of the healthy cells that are morphologically the same as the healthy cells, but they have this slight metabolomic change that in the future maybe can be transformed into the cancer cells. But that's not all. We are also currently analyzing all of this metabolomic data to find the possible new tumor markers. And as you can imagine, each of these spot of this image represents one MS spectra. So the amount of data that we obtained is extreme, and the data analysis will take time. But what we hope to do with all of these results is that they will still aid us in not only the design of the drug delivery system with the optimal tumor penetration, but as well the therapeutic outcomes, but also in the development of preclinical models that will mimic much better the human tumors. And finally, as a conclusion, I would just like to say that all of these data are just the beginning. As you can see, we just had 2 projects with the multi-MS imaging, but that we are sure that this technique has a great potential to be a technique that will help us elucidate much better the in vivo fate of our drug delivery systems in the future. So at the end, I would just like to thank all my team, my mentor, Maria Jesus Vicente, for investing in analytical chemistry in nanomedicine, the use of analytical chemistry in nanomedicine, or for investing in me as an analytical chemist, but also to Professor Malcolm Clench from Sheffield Hallam University, Julia Lorenzo for providing us the glioblastoma in vivo model, and Professor Ron and Michiel for backing up my idea, for believing in this project, in the technique and using nanomedicine, because without their support with the latest and the most advanced MS imaging facility in the world, this project would just not be possible. So at the end, I know that we have this question and answer session, but in case that there are some people that didn't get the opportunity to ask me something, or if you just want to connect with me, please scan the QR code, and where you will just access an online form where you can ask me anything that you want, or just to connect with me, as I said, and I will be glad to take you on our MS imaging journey from our humble beginnings to the exciting future that I'm sure it lies ahead.
Many thanks, Snežana, for the nice talk. And actually, there are a lot of results, a lot of work. So thank you for bringing these results to your presentation. Thank you so much. Okay, so let's go directly to the questions of our participants. The first one, what could be the ways for novel applications of MS-based analysis, perhaps improvement and development of new instrumentation and techniques, bioinformatics tools? What is your opinion?
Definitely bioinformatics, definitely the software, and even though that with the Bruker software skills, it's already developed nicely and it's user-friendly as well, but the bioinformatics, of course. But one of the things that I was already talking with Professor Ron, and they were already working on that, is the spatial resolution, to improve the spatial resolution, because as you can see in my case, we did use 10 and the 30 micrometer resolution as the lowest with this instrument. With some of them you can use until 5 micrometer, but you lose a little bit of sensitivity. So right now there is, in the future, I think that it's definitely we can improve more the spatial resolution, and at the same time to get to even the subcellular resolution with the MS imaging. Yeah.
Many thanks, Snežana. So we have Zaida. Maybe I can see your hand here. Maybe you can ask your question directly to Snežana. Can you hear us? No.
She needs to unmute, or
yes. No, maybe Daniil also raised his hand. Maybe you can ask. Hi.
Yeah, good afternoon.
Good afternoon.
My name is Daniil, and I have a question regarding the data processing, which you told you used spraying. And could you tell me, please, why didn't you use sublimation, which was proven to be less delocalized sample preparation protocol? Maybe there is some kind of thing behind it. Well, describe it. Don't you think it will be a little bit
in our case, what we used is an HDX sprayer. One of the sprayer that it's currently is used a lot in the MALDI, and one of the most advanced, let's say, sprayer. And with that sprayer, that it's already the protocols that were performed, that were developed as well in the Sheffield Hallam and at Maastricht University, it's proven that it's with the spraying, you have directly the evaporation droplets that are coming, evaporated droplets of your matrix solution that goes onto your sample. So with that one, it's not like the previous and just the spraying technique that we were using before, and that you can actually have more delocalization. In this case, it's much more controlled. So you have, in this instrument, you have a nozzle that is heated to a really high temperature, meaning that at the end you have the droplets of the matrix solution that it's evaporated already. So with that one, you remove this, let's say, the bad thing of the before techniques that is the spraying. So sublimization, I didn't use it so far. I know that in the Maastricht University, they use it for some cases, for some things, but for me, I found this one that it didn't get to the localization, the metabolites.
Okay, thank you. This is
You're welcome.
Very describing. And the second question is regarding SEALS data analysis. So you just described the SEALS data analysis. Could you describe more which steps have you included into it? And similar to this question is about the segmentation on 24th slide, how it was performed, which methods have you used, and yeah, that's it.
Yeah, well, when it comes, and there is not enough time, it's never enough time to explain everything that we want. That's why we have these questions at the end session. Well,
we can discuss it by the QR code if you wanted to. And
yeah, that's also, but I would just like, I mean, this is a really important as well question. So thank you for asking it. But one of the things when you are analyzing the multi-data and when it gets to the imaging part is the normalization that you have to perform, because once you have, as you can imagine, and spraying the matrix solution, the matrix on the top of your samples, you can have a different, you can in order to remove the possibility of interfering of the matrix as well, you can use the normalization of your samples. Let's say there is a standard solution that you can use and to normalize all of your MS spectra. Or in my case, I found that it's actually the matrix ion can be used as a normalization. So each spectra and each let's say each spot that we have of the 10 micrometers is actually normalized first with the matrix ion, and then so that we can make sure that we can compare the different pixels. Let's say that I hope that I answered it well. When it comes to the segmentation part, it's the let's say the segmentation of all of the samples and all of the tissues. We first, again, it was always the normalization, and then afterwards there is a part in the skills that it's a really user-friendly, that you can just select the part that you want to segment, it's a part of the tissue, and then you can, at the end, it has Michiel said, play around with the software and try to find compare. As you can see, I compared the MRI images with the MALD image and try to see if I can overlap them and mainly see the tumor that's differentiating it from the healthy tissues. And then you can continue with the data analysis. But please scan the QR code that we can discuss about this much more in details afterwards.
Thank you very much for your answers.
Thank you.
Thank you, Daniel, for the nice questions. So let's go to the next one. Which is more general, I guess. What is the role of artificial intelligence and machine learning in MS?
Right now, again, as we were discussing about the bioinformatics, that is one of the things that I was thinking about, definitely that we can improve that. And with the data and image analysis when it comes with artificial intelligence. But apart from that, there is also the, let's say, the reconstruction of the images. And as you can see with the multi, we can analyze just one section, okay? So when it comes to the tumor tissue, the tissues or the brain tissue, it's not just one section. It's a 3D organ. So at the end, when it comes to the artificial intelligence, it can help us when we analyze different sections of the brain tissue to reconstruct as well the 3D model of the brain. So on that sense, yes, for sure. I hope that someone is doing that. Yes.
There are more things to do there. So, Snežana, may I ask you a question of mine now? So how many polymer therapeutics are in clinical use today? So there are actual drugs there.
I mean, when it comes to the polymer, between 25 and 30 are already in the clinics, around like that.
Okay.
Not as much as we would like to. That's for sure. That's for sure. But one of the things that we are implementing, and as you can see, Maria Jesus started in the last, even before me coming, to implement much more the characterization part and involve analytical chemistry in all of this process. Because what we saw that it's at the end, when it comes to the clinical parts and when we do the clinical studies, there are a lot of candidates that don't go through because they were not characterized well. So with this, there are a lot of many people, not just Maria Jesus, that it's investing in analytical chemistry, and not just having analytical chemists us in the companies, but also in the lab. In the labs, we have things to say. I know that my colleagues will say the same. So yes, and I hope that in the future we will have many more.
This is great. Okay, another question. Can you summarize the steps to create bioresponsive polymer drugs linkers that allow controlled drug release at the site of action?
Oh. Yeah,
it's a lot, right?
Yeah, no, it's mostly because I mean I love protocols, as you can see, and I love to have schemes, and I love to have everything to be organized and everything to be logical. But you cannot have basic. I mean, we can have a basic scheme, and we know that we can have some things that are really important. For example, we need to know it's not just enough for us as a chemist to know the chemistry and to connect these linkers, the drug with the polymer. We also need to know the biology. We need to know the target that we are using. So first thing first is the target. We need to characterize well the models. We need to characterize well the tumors. We need to know if there is a high GSH content in this case, or the presence of some other enzymes, for example, the enzymes that will cleave the peptide bonds. So we need to know first that. Then we need to see the structure of our drugs, because if they don't have the functional groups that are able to connect to the polymer, then we cannot conjugate it. Okay, so that's the second part. Then when we have those two parts connected, those two parts resolved, we can start by synthesizing the new polymer drug conjugate. Once we synthesize, then we usually synthesize it with the different carriers. Okay, so we have different candidates. Once we have those different candidates, we characterize them. We do the in vitro studies, and from there, those toxicity studies, we see which one is working and why is it working. So once we know that, we go again to the beginning. So it's kind of like a circle. We go again to the beginning and we go back to the synthesis, and we change something in the synthesis part, or the drug loading. Is it the drug loading that is affecting? So it's, let's say, a circle process. But that is some basics that I can cover, but it will definitely depend on the case-by-case basis.
Exactly. And you always have to ask the right questions, right, in each step. Yes. So what is going wrong? What is going well?
Yeah. And when it's not going wrong, to understand why is it not working, let's say like that. And once we understand that, we can change some things, and not just I always say that for me it's interesting when something is not going well, because there is something to answer. There is a question that you should answer to it. But if everything goes well, it's just like
you can never learn.
No, exactly.
Thank you, Snežana. So we have time for one more question quickly. What are the advantages and disadvantages of therapeutic polymers?
Okay, when it comes to the therapeutic polymers, is it the polymer therapeutics or is it the polymer drug conjugates that we are using? Because inside the polymer therapeutics, we do have different groups. So I would focus now on the polymer drug conjugates that we are working on. And one of the greatest advantage for this group is that we can make sure that the drug that is conjugated to this carrier is actually arriving to the place with the carrier. While in the case, for example, of the capsulation of the drugs inside the polymer or inside the micelle, we cannot be so sure that the drug will arrive to the place of action, because there is less stability in the bloodstream compared to this chemically bonded the drug to the polypeptide or the polymer. And the second advantage is also as well that we can control the release. We can control the release. We can control the total drug loading. So there are, let's say, many advantages but that at the same time complicate more stuff. Let's say it like that. So you have more steps to synthesize it, so which means that you need to control much more all of the steps of the synthesis, and then at the same time, when it gets to the site of action, you need to control to make sure that it's releasing, as you saw the examples of our studies. So we need to characterize them much more. So let's say it. So I would say that as a disadvantage, more steps.
Great. I think we answered this question. At this point, Snežana, I would like to thank you very much for being here with us today. I hope that we will have the opportunity in the near future to talk more about this interesting subject. I think this webinar was really interactive with questions and answers. So thank you very much for this webinar.
Thank you very much, you, for organizing, and everyone for being here and listening.
Many thanks.
Many thanks. Bye.
Bye. So, yeah, we are about to finish, to complete our webinar. So I would like to remind you a few things about EU-OPENSCREEN. So you can see here that you can visit us online and use our website to navigate in our different services, or contact us if you have a training question at this email address. You can also sign up for our newsletter here, and you can learn more about our training activities in the future. Thank you for being here today, and I hope you had a nice time with us. Thank you very much.